Alright, we're back. And as I said, this is going to be a quick one. And the topic is continuing cell division. It's going to be mitosis. So I don't have to do a lot in this because you have your textbook, you have animations and simulations. So what you do is just Google mitosis or cell division animations and simulations. And instead of being static images like these, they'll be moving around and there'll be a narrative and you can stop it. Okay? So this is kind of amazing because we mentioned in the cell cycle, cells start off as one cell and then they replicate and give you two. And mitosis has certain phases. I quickly went through that. They were prophase, meta, ana, and tele. So prophase, metaphase, anaphase, and telophase. So you would want to know those different phases. And it's not that hard, even though you see all these pictures up here, because each one has something that's very characteristic of it, okay? And I will point it out on this one right here, okay? So this one is pro, meta, ana, and tele. So over here, this is a prophase. And how you know it is you can start seeing chromosomes. There are these things that look like little bow ties. Those are replicated chromosomes. And you may remember from lab or reading your book, a replicated chromosome is called a chromatid, and they look like little bow ties because in the middle, the replicated chromosomes are connected by a special structure called a centromere. So this is prophase, okay? After prophase comes metaphase. What I do is I think meta begins with the letter M, you can think middle. And in metaphase, the chromosomes line up in the middle. See, like that. That's in the middle. So 1, 2, 3, they're migrated. So this is a sequence. It looks like this, then it starts to look like this, look, and then it's frankly in the middle, and then it's really in the middle right here, okay? So prophase, they kind of look like this. You can see chromosomes, they're not quite organized in the cell, like relative to the ends or middle. Metaphase, they are sure enough in the middle. And then anaphase, they're like little bunches of bananas or V's headed in opposite directions. And that's like right here, see? So prophase, metaphase, anaphase when they're going to opposite ends, and then you have telophase. So you have a nucleus here, and we've divided the nuclear material. That's called karyokinesis. So mitosis has two things that have to happen. Divide the chromosomes, aka the DNA, and then divide the cell itself. The cell itself is divided in what's called telophase, in right here. And notice it looks like you're pinching it apart, like you get a dinner roll and pull it into two parts. The cell is constricting. So what's interesting is that the constriction is occurring right here. It's not occurring here and here. If the constriction to divide it occurred here and here, this would be really messed up, okay? And whenever cells get messed up, then we get messed up, okay? So what's really great is the cell goes through this in a certain manner, very predictably, and then you end up with two cells. So if you remember from my cell cycle, we start with one big cell and then there was two little cells, and each one of these, depending upon what they do, they will go through the process again. All cells don't go through mitosis as is generally thought. There are two categories of cells that don't do this, but maybe under certain special conditions. They are muscle cells and nerve cells. They are referred to as being post-mitotic. Post is after. They don't do mitosis. But as more research comes about and things are discovered, you find out, sometimes muscle cells and nerve cells could reproduce. Now, wouldn't that be great, especially for nerve cells? Because when persons have brain injury or have some kind of disease, then the cells that are bad could be replaced. Okay? That also brings in stem cells again. This is pretty good. And here's another one, mitosis and cytokinesis. Okay? So, in the jargon, the mitosis could be, we're only interested in the DNA and that. And cell division is the whole thing. So karyokinesis is when you're dividing that, cytokinesis, but typically it's all called mitosis, but I think you have it. And again, what you see, what phase is this? Can you see where they are? Where are the chromosomes? What part of the cell? Are they scattered at random? Are they at ends or are they in the middle? Yeah, they're in the middle. If they're in the middle, what phase is that? What phase begins with the letter M? Metaphase. So on one side of metaphase is prophase, and on the other side is anaphase, where they migrate. And then you have telophase. This looks like it's kind of missing one. This is in the middle, and you don't really see the anaphase. like here. And we see this. So this set of diagrams looks like it shows us prophase, metaphase. It really doesn't show us anaphase because the chromosomes have ends. So that's really telophase, and then it shows the two daughter cells. I don't know why this chart does that, okay? So these are chromosomes moving around. They're like hot dogs. You can see them? So what you're really trying to do when you move your chromosomes is you're moving DNA. So here's a chromosome. Remember I said it looks like the letter X or a bow tie is connected. So these are replicated chromosomes. They are identical. They're called sister chromatids. They're connected by a special structure called the centromere. So if you take a chromosome and pull it apart, unravel it, you come down to the DNA helix right here. So it's quite amazing. For instance, if you go on a trip or you buy something and there's a cord wrapped in with electronic devices, have you ever been able to put it back in it like it was from the manufacturer? I know I never have. And when you have all these clothes and you want to get them in the suitcase, you can't just stuff them in. You have to take them out and fold them and, pack them and do that. That's what happens with chromosomes, because the amount of DNA in a human cell, one human cell will be as tall as I am. So you take that much DNA and you coil it, fold it, stick on proteins, and make these things called chromosomes so that the cell can more easily move it around. If it just had a thread of DNA, okay? So this is showing DNA, and what's really weird, as I tell students, is this is what makes you, okay? Now, there's another process that we're not going to go into called meiosis. So there's mitosis and meiosis. Sometimes say meiosis, mitosis and meiosis. They are two cell division processes, but they have totally different objectives. With mitosis, you have one cell with 46 chromosomes, and your objective is to produce two new cells from that with 46. In meiosis, you start off with one cell with 46, and you're going to get cells that have half of that number, 23. Now, why would you need to cut chromosome number in half? Why would you ever need to take a cell that has 46 and make 23? Well, that's it. Reproduction. You have egg and sperm. They show up through meiosis. They have 23, so when an egg and a sperm come together, let's add it up. 23 and 23 is 46, so you have now reconstituted the chromosome number. And meiosis has two cell division processes that we're not going to go into this video. I suggest you look at an animation of mitosis and an animation of meiosis and just go back and forth and compare them. In mitosis, you run through this process one time. Pro-meta-anatellum, it's a wrap, it's over. But in meiosis, you run through it once and then you do it again. Okay? So there are two cell division processes. They still use the name pro-meta-anatellum, but it's prophase 1, metaphase 1, anaphase, telophase 1, and then you get into 2. Okay? But the best way to get that is look at your book and look at an animation of it. Okay? I think what's amazing is that you are an assemblage of chromosomes, half from your mom and half from your dad. Okay? So some kind of way, and the chromosomes have numbers. They have 46, 23 pairs. So you can talk in terms of chromosome number 1, 2, 3, 4, 5, 6, right to 23. So what happens is, in the process of producing egg and sperm, your mom has a chromosome number 1 from her mom and her dad. They come together and then they divide. So chromosome number 1 can find chromosome number 1. 2, 2, 3, 3, 4, 5. If one lined up with 5 or whatever number, it would be a problem, okay? Also, how many chromosomes do you need to have? 46. If you have too few, too many, then it's a problem, okay? And there are different conditions where you have that. One that you may have heard of is Down syndrome. So one presentation of Down syndrome is when you have trisomy 21, chromosome number 21. It's in 3 copies, trisomy, okay? But there are some other ways to get it also. When we look at this one, chromosomes have a beginning part and an end part. And if you have cells, like cells replicate, okay? If you give them everything they need, opera music, give them snacks or whatever, they don't have cable TV, they will reproduce, go through the cell cycle only so many times. And after that, it's a wrap, okay? Part of that is that at the end of chromosomes are some special pieces of material called telomeres. and the telomeres keep the chromosome from deteriorating. So every time cells replicate, they get a little bit of damage, especially at the ends, okay? But if they have telomeres that stay intact, then the chromosome stays intact. And by the way, here's a friend right here, a turtle. Now, turtles live quite a while unless someone steps on them or runs over them, okay? And it's kind of thought if you don't kill a turtle might live forever. And what they found is that turtles have very good telomere presence. That's an enzyme that causes them to be formed called telomerase. So turtles are pretty special because they can protect the ends of their chromosomes, and they can live for a very long time. And again, it's thought if you don't kill a turtle will just live forever. Also, here's some little friends that I have, and of course I like turtles, so here's another turtle. Hello, there's a turtle. And here is fish. Okay. And here is a butterfly. So we have fish, turtle, I use finger puppet, and we have a butterfly. These are distinct organisms. Like you would never mistake a fish to be a turtle or a butterfly. Well, what makes a fish, a turtle, and a butterfly is their DNA. And that goes back to this. It's really wacky. Some kind of way, this determines everything about you and what different organisms on the planet are, what they are. It's this DNA. Okay? And in the earlier chapters, we learned about DNA and the nucleotides, ATCGs, and you can see it right here. Maybe you can zoom in and see it. That's an ATCG. Oh, excuse me, let me turn this off. Bad cell phone, okay? So what happens is that these sequences of letters, and it's in the earlier chapter in your book, okay? And what's really weird is this sequence of ATCGs gives you what you call your DNA sequence, it gives you genes, and it makes you what you are. So you kind of have to accept it. I've studied it, I like it, and I've accepted it. When I think about it, it's really crazy. Now here's a butterfly, and this is pretty amazing because you know what a butterfly looks like. And it starts off as a caterpillar. A caterpillar looks nothing like this. Okay? So what happens is, of the DNA in that organism, some of it's turned on to say, let's make a caterpillar. And then at some point, it turns off and let's make a butterfly. And then it goes, metamorphosis, which is kind of amazing. Because a butterfly and a caterpillar look nothing alike. See? So that's pretty darn amazing. And what's interesting is that the mechanisms that control what these things look like that's a head, that's a back, inside, top, bottom, same thing with you, same thing with this. It's true. The genes that control what goes where is pretty common in different life forms. What that means is you can study fish and learn something about humans. You can study this and learn something about humans. And of course, turtles from the telomeres, that's it. So life is more common than it's different, but we see turtles and giraffes and cockroaches and all that. But when you get down to it, it's more similar than it is different, okay? Everything's trying to do the same thing. It's a hot day outside. It's a cold day, you see? How do you deal with it in different organisms? Deal with it in different ways. So this is mitosis, and I gave you an intro to meiosis and the significance of it, okay? And I think that's pretty good. So I'm going to stop and say goodbye, and